Let's talk about Watson and Crick's model of DNA. In order to understand this model, we must first discuss the components. Nucleotides are the building blocks of DNA, and they're composed of three things: nitrogenous bases like adenine, guanine, thymine, and cytosine, a sugar, and a phosphate group. The four bases fit into two different structural categories: purines and pyrimidines. Purines are double-ring structures, and pyrimidines are single-ringed. In DNA, the purines are adenine and guanine, and the pyrimidines are cytosine and thymine. There are smaller compositional differences within the categories to differentiate these bases. Chargaff's base composition studies discovered that in DNA, the amount of adenine and thymine is proportional, and so were guanine and cytosine. However, the two pairs were not always in the same concentration as each other. This suggested base pairing between adenine and thymine and between guanine and cytosine. The purines pair perfectly with their respective pyrimidine counterpart. Adenine and thymine line up to create 2 hydrogen bonds, while cytosine and guanine make 3. Hydrogen bonds are relatively weak interactions between differently charged molecules and are not enough to stabilize the structure entirely. The next component in a nucleotide is the sugar. This is ribose in RNA and deoxyribose in DNA. They have a cyclical structure with 5 carbons total, and the carbons are labeled 1' to 5', and the order is crucial for the way the nucleotide is structured. You can tell the difference between deoxyribose and ribose by looking at the 2' carbon. If the 2' carbon has an OH bonded to it, it is ribose, whereas deoxyribose is missing that oxygen, hence the name, and only has an H. The final component in a nucleotide is the phosphate group. This phosphate group consists of 4 oxygens that are bonded with a phosphorus. 3 of the oxygens have a negative charge, making the group negatively charged overall. To form the nucleotide, let's go back to the sugar. The 2' carbon in DNA only has an H because it is deoxyribose. The 1' carbon attaches to the base, A, T, G, or C, and the 5' carbon attaches to the phosphate group. The empty 3' carbon attaches to the phosphate group of another nucleotide, and this is where DNA starts to form. The phosphate group bonding to the sugar of another nucleotide forms a phosphodiester bond. The phosphodiester bonds are stronger than the hydrogen bonds and help form the phosphate backbone of DNA. So now you have the hydrophilic backbone on the outside of the helix and the bonded hydrophobic nitrogenous bases on the inside of the DNA. The strands are antiparallel, so they run in opposite directions. The directions are based off of the orientation of the sugar molecule, so the 5' carbon points towards the 5' end and the 3' carbon points towards the 3' end. The bases pair in a way that their sugar molecules are opposite to each other, and the bases themselves are perpendicular to the backbone. The perpendicular orientation is called base stacking, which maximizes hydrophobic interactions to keep out water, and this is the main stabilizing force in DNA. When DNA is twisted up, it has a major groove and a minor groove. This refers to the distance between the phosphate backbone. Where the backbone is closer together is the minor groove, and where they are farther apart is the major groove. Handedness refers to the twist direction, which is determined by the orientation of the backbone. If the backbone is pointed up to the right, it is right-handed, and if it is up to the left, it is left-handed. Normal DNA is right-handed. TM is the midpoint of thermal denaturation in DNA, where DNA is 50% denatured, so 50% of the strand is separated due to H bonds breaking. If you take a sample of DNA and place it in a high concentration salt solution, positively charged sodium ions partially neutralize or shield the negative phosphate backbone, making it less polar and therefore less hydrophilic, causing it to dissociate from water. This makes the DNA strands compact and become more stable, so it is more difficult to separate the DNA strands. In order to separate these strands, more heat is required, so the TM would be greater.